All right. Hello, everyone. Um, Today we're going to be continuing where we left off last time. Um, we're going to be finally finishing chapter one of the cell. Um, it's been a long time. We have these 40 cards that we just need to speed through. It's another late night stream. Um, we're just literally all we have to do is finish off these last 44 cards, and then look, I'm literally going to change the card settings just so we can look at the rest of the so we can look at the rest of the look 40. Oh wait, hold up, we need to actually, I think we might need to change the sub deck. It doesn't even matter if I change that. Hold up. Okay, so we have 44. This one we already know is going to be long. There's like there's like several processes there, like that's to be expected. 82, 100, which should be easy, bro. If I struggle with the nervous system, I'm actually gonna break. I might have a breakdown, bro. I'm not gonna lie to you. Endocrine system, that one might be torturous. I'm not gonna lie. Respiratory system, maybe. Cardiovascular, I don't know. There are definitely some cardiovascular, immune, digestive, like all those I already know are gonna be somewhat difficult. Homeostasis, it's like okay. Um, and this one, look at that. Look at the beauty of biochemistry. Ugh, I just need to. I finished two. I didn't know. I actually didn't know that I finished two chapters in biochemistry. Okay, I forgot it. I forgot I did that. But go me. So, um, all right. Well, let's not do 700 cards today. Uh, while that would be lovely if I had the brain capacity for that, or the godly ability to do something like that. Um, a person that is capable of finishing 600 Jack Sparrow cards in one day, like, yo, imagine that would be. They would finish the deck in literally 10 days, bro. That would be. You know how immaculate of a thing that would be, and to actually remember all 600. I can't even imagine what the reviews would be like. I would have to give myself like a week break. Let's say I went that hard. I would need to give myself a week break just to recover from 600 cards. 600 cards, and then the following reviews. Oh hell no. That sounds like actual torture. Sitting through the reviews every day is like a full time job. Um, what are the two types of bacterial cell wall? This is gram positive and gram negative. These first couple cards are gonna be nice and easy. We're just gonna cruise right through them. Oh, lo-fi. I got you guys. I'm not forget. It might take me a couple minutes to remember to put it on, but I'll remember. Let's see if we can get. I shouldn't be testing things out on stream, but I'm gonna look it up. Uh, and we're also gonna mute the audio because I know that if I don't mute the audio, then you guys are gonna hear it, and somehow I'm gonna get a copyright strike. So, hold up. Let me let me see if there's anything like really long and like. We're gonna. I need to put. I need to put that in quotes because if I don't put it in quotes, All right, we got a new lo-fi track for today. Um, all right, now we can actually get started. Have our little music in the background. We back to the stick drift. You know, it's a it's a casual Thursday. Well, Friday now. All right, I remember that. I'm gonna remember that. What are the two functions of the mitochondria? I don't know. Produce useful chemical energy via the citric acid cycle and ETC, and induce apoptosis by releasing ETC enzymes. I didn't know that. That's cool. What are the three types of layering found? Like, I vaguely remember that, but not anywhere close to enough to. I would never bring that up in conversation. What are the three types of layering found in epithelial cells? I have no clue. Oh, that's what it was asking. What are the three types of anaerobes? Um, that's going to be obligate, facultative, and aerotolerant. We're just gonna skip right through that. Like we just easy those because it's like we learned it already. What are the three structural components of the cytoskeleton? Um, we have the microfilaments, the microtubules, and the intermediate filaments. We easy those. We easy those because we know them. We know them, bro. What are the three shapes of the epithelium? Cuboidal, squamous, and Wait, columnar. Columnal? No, columnar. Yeah. That was a little hard. I'll give it hard. What are three shapes of bacilli, spirali, and uh uh oh uh Kochi? um we'll give it hard. Like it did take me a second. It produces ATP and it can induce apoptosis by releasing ETC enzymes. What are three types of layering found in epithelial cells? Um, three types of layering found in epithelial cells. Uh, Stratified, simple, and pseudo-stratified. Three places an endosome can transport material to. Um, it's going to be like the ER, the nucleus, and the membrane. Oh, okay. The membrane, the lysosomes, and the trans-Golgi. What are three forms of genetic recombination? So we have transformation, transduction, and last one, transformation, transduction. There's a lot of lists in today's deck of cards, bro. Transformation, transduction, and uh, transformation, transduction, and conjugation. That was really hard. What are the three domains of life? Oh, this is gonna be this is like what is it like prokaryotes, eukaryotes, and uh or is that too broad? No. Is it archaea? Damn. Something like that. Like that was pretty close, right? Like bacteria, eukarya, and archaea. Okay. So we have lysosomes. Um, I don't know if you guys can tell, but that rest day was clearly much needed. Um, we have eukarya, eukarya. Wait, no, what? That's not even the question. It can transport it to the lysosomes, the trans Golgi, and to the membrane. To the membrane. That's pretty hard. The three components are the filament, the hook, and the basal body. What are the tenets of cell theory? Um, all living things are made up of cells. All cells come from other cells. Uh, uh, I don't know how many more I got in me. Uh, I don't know. Yo, that was pretty. I got the first two. I didn't expect to get them in order. Cells carry genetic information in the form of DNA. Okay. Okay. We have eukarya, bacteria, and archaea. The functions of the peroxisome. Okay, this is gonna be too specific for me, but it's just gonna. It probably breaks stuff down. Break down fatty acid chains and beta oxidation. Synthesis of phospholipids and some contain some enzymes for the pentose phosphate pathway. Okay, so breakdown of fatty acid chains, um, breakdown of fatty acid chains. I already forgot. Synthesis of phospholipids, breakdown of fatty acid chains, phospholipid synthesis, and as it contains some of the enzymes for the pentose phosphate pathway. Fatty acid chain breakdown, phospholipid synthesis, and some of the enzymes for the pentose phosphate pathway. Okay. What are the functions of the epithelial tissues? Um. Okay. So it covers the body, lines its cavities, protects against infection and desiccation, but it can be involved in absorption, secretion, and sensation. Okay. Okay. I'm put a like a small cue in the question. Um. Okay. So all living things are made of cells. All cells come from pre-existing cells, and cells pass down genetic information in the form of DNA. Yeah, pretty much. What are the functions of the peroxisome? So this is fatty acids breakdown, phospholipid synthesis, and it contains some of the enzymes for the pentose phosphate pathway. What are the functions of the centriole? It works in mitosis. Oh, it is an MTOC, a microtubule organizing sensor, and for separation of chromosomes during mitosis. Okay. What are the three shapes of epithelium? Squamous, cuboidal, and columnar. What are the three shapes? So we have bacilli, spirali, and cocci. So epithelial tissues. So they cover the body and they line the cavities. Um, uh, they can be involved in absorption, secretion, and I don't know what else. Absorption, secretion. Um, I'm not sure. Providing protection against infection and desiccation. Provide protection against infection and desiccation. Okay. So it's a microtubule organizing center, and it's crucial in aiding chromosome separation during mitosis. Three forms of genetic recombination seen in bacteria. Um, we have three forms of genetic recombination seen in bacteria. So we have transduction, transformation, and conjugation. The four tissue types. Uh, I don't think I know this. Um, I mean, like besides like epithelial and connective. Oh, muscle nervous, bruh. What are the three components of bacterial flagella? Um, the basal body, the hook, and the filament. What are the functions of the epithelial tissues? So they cover the body and they line the cavities. Let me turn down the volume for myself a little bit. Um, um, so they provide protection against desiccation and something else, and they also are involved in absorption, secretion, and something else. Sensation, infection, and desiccation. Okay. To the lysosome, to the membrane, and to the trans-Golgi network. Positive and negative sense. No, what? No. Gram positive and gram negative. The three domains of life: eukarya, prokarya, eukarya, bacteria, and archaea. All right. The four tissue types are um, epithelial, connective, nervous, and muscle. The four steps of a gram stain. I don't know this. Stain bacteria with crystal violet, then add iodide. Um, okay, so you stain the bacteria with crystal violet, and then you add iodide. So the iodide is gonna bind to the crystal violet and trap it in the cell. So stain it with crystal violet, then add iodide, and that's gonna work with um, crystal violet is gonna apply to gram positive. So apply, stain the bacterium with crystal violet, add iodide, and then you're gonna you're gonna wash it away with ethanol to disrupt the ethanol is going to be used to disrupt the membrane and to wash out the crystal violet from the gram negatives, and then you're going to add safranin as a counter stain. Okay, hopefully I remember that. What are the three types of layering found in epithelial cells? um Is this just simple stratified, pseudo stratified? Yep. So it's gonna cover the body, and line the cavities. Um, it's gonna provide protection against infection and desiccation, and it's going to be involved in absorption, secretion, and damn, I can never get this last one. Sensation. What are so all living things are made of cells. Cells come from pre-existing cells, and last one, cells pass down genetic information in the form of DNA. I have to keep putting hard until I get it. No trouble. So it creates chemical energy in the form of ATP, and it also is like it can induce apoptosis by releasing its ETC enzymes. What are the functions of the peroxisome? So using its hydrogen peroxide, it can perform breakdown of fatty acid chains. It can synthesize phospholipids and synthesize phospholipids and synthesize phospholipids. I don't know. Okay, I didn't even read it. Contains some enzymes for the pentose phosphate pathway. I was not going to get that. What are the four steps of the gram stain? There's not even the four steps in doing it. Like not, it's just simply not four steps. Um, so um, it's you're gonna stain with counterviolet, not counterviolet, crystal violet first. Um, and then you're gonna add iodide, which is a which is gonna trap it in the cell. Um, and then you're gonna add ethanol to wash it away from the gram negative, because the gram positive are gonna have a thick layer of peptidic glycan, so it's not gonna wash away. But it will wash away for the gram negative that have that thin layer. Um, so then it's gonna be you're gonna add in safranin as a counter stain. What are the functions? I'm gonna click again just because it's I feel like it's not accurate. What are the functions of the centriole? Um, what are the functions of the centriole? It's a microtubule organizing center, and it's also crucial in separating chromosomes in mitosis. What are the functions of the epithelial tissues? Um, so it's gonna line the cavities. It's gonna cover the body, provide protection against desiccation and infection, and it's involved in absorption, secretion, and sensation. Okay. What are the functions of the peroxisome? So it's fatty acid chains breakdown, phospholipid synthesis, and it contains some of the enzymes for the pentose phosphate pathway? What are the three places an endosome can transport material to? Um, Three places an endosome can transport material to: the lysosome, the membrane, and the trans-Golgi network. Um, Eukarya, bacteria, and archaea. What are the four tissue types? Epithelial, connective, nervous, and muscle. What are the four steps of a gram stain? Um, what are the four steps of a gram stain? Um, uh, what are the four steps of a gram stain? Okay, so you're gonna add crystal violet first. Um, you're gonna add crystal violet, then you're gonna add iodide, right? And then after that, you're gonna wash it with ethanol. And the reason why you're washing it with ethanol is because the gram positive, they have this thick layer of peptidoglycan that it's gonna hold it in. It's gonna hold in that stain. So when you wash the gram negative bacteria with the ethanol, it's gonna wash away the peptidoglycan and the crystal violet together. So then when you apply the safranin, that counterstain, then you obviously have that that's the distinguishing factor, like because they're different colors. What are the three shapes of bacteria? Cold sheep, bacilli, and spirali. Cuboidal, columnar, and squamous. Three forms of genetic recombination: transduction, conjugation, and transformation. What are the tenets of cell theory? Um, uh, so all living things come from cells. All cells come from pre-existing cells, and cells pass down their genetic information in the form of DNA. What are the three components of bacterial flagella? Um, it is the basal body, the hook, and the filament. What are the four stages of bacterial growth? Lag, log, stationary, and death. Okay, I'm trying to figure out what the color code is for you guys because I've realized that I'm using my controller so you guys can't see which buttons I'm clicking, but like it's not as easy as like it seems. Like these are obviously the new cards. These are ones that I get that are like it's not necessarily wrong. It's just like I didn't click easy. If it moves on to the next day, it marks it as green. So yeah, that's that's literally the best that I can give. Like if I move on to the next day, but I'm trying to figure out what the color code is besides that. I don't know if it like the smaller the line is, I don't know if it I don't I don't know. I haven't figured that out yet. What are the components of viruses? You have the tail sheath and the tail fiber. Oh damn. The tail yeah. I think so. Um the genetic material, the protein coat, or the and or the envelope. Okay. So it's gonna cover the body and line the cavities. It's gonna protect protect you from desiccation slash infection, and it's gonna be involved in absorption, secretion, or what's one last thing? The one last thing: absorption, secretion, and sensation. The functions of the central, it's the microtubule organizing center, and it's also crucial in the uh, separation of chromosomes during mitosis. What are the characteristics of microtubules? Okay, this is when things start getting annoying. Hollow polymers of tubulin, um, track for kinesin, and there we go. I like that. I like that question better. I feel like it's more specific. Um, what are the functions of the peroxisome? The peroxisome, it functions in the breakdown of fatty acid chains, the synthesis of phospholipids, and it contains some of the enzymes for the pentose phosphate pathway. What are the components of viruses? The genetic material and the protein coat slash caspid, um, or capsid, not caspid. Um, and the envelope is the option one. The characteristics of microfilaments. Okay. Uh, See, because the thing is, I just can't. Like, the thing is that this is supposed to be the broken down version, and it's like really not. Like, I don't like essays. I don't like essay flashcards. So, like, you might as well just ask me all these as separate cards, and I genuinely might enjoy it more. What are microtubules slash? What are microtubules? Microtubules are hollow structures of Um, Microtubules are hollow structures made out of tubulin. The motor proteins are kinesin and dynein. They're used in the cilia and flagella, as well as what's it called? Um, as well as in centrioles during like mitosis. Hollow polymers of I don't know what I said. I don't know if I said polymers, so I'm gonna say it again. So you you stain the bacteria with crystal violet, then you wash it with no, not yet. You don't wash it yet. You put the iodide, add an iodide, and then you wash it with ethanol because the iodide is going to lock it into the thick peptide glycan. But the thing is that because there's a thin layer in the gram negative, what you're going to end up doing is washing away the peptide glycan layer altogether with the crystal violet. So then you add in the safranin after, and you have like the distinguishing your main like distinguishing factor between gram positive and gram negative. Um, the lysosome, the plasma membrane, and lysosome, plasma membrane, uh, the lysosome, the plasma membrane, and the trans Golgi network. Microfilaments are, I don't know, they're like rope-like structures made out of actin. Um, uh, it's motor protein. Damn, I don't remember a lot of this. Okay, it's organized into bundles slash networks and provide compression resistance. Can generate force by interacting with myosin. Important for cleavage furrow during division. Okay. What are the four tissue types? Um, Epithelial, connective, nervous, and muscle. The three domains of life: eukarya, bacteria, and archaea. Microtubules are hollow, hollow. What is the word, bruh? It's not structures. Hollow polymers. Okay. What are the functions of the epithelial tissues? It's gonna be um, so it's gonna line the cavities. It covers the body. It provides protection against desiccation and infection, and it's going to uh, and be involved in absorption, secretion, and sensation. What are microfilaments? Microfilaments are damn. It's like, bro. It's like, once again, they're like these rope-like structures that are organized. It's made of actin. It's organized into networks slash bundles to resist compressive force. The motor protein is myosin. It interacts with myosin, and using that by doing that, it can generate force. It's important for the formation of the cleavage furrow during mitosis. polymerized actin filaments. Okay. So we have the genetic information and the protein coat or capsid, and then the envelope is optional. What are microtubules? They're hollow again, bro. Hollow hollow polymers of tubulin. Motor proteins are dynein and kinesin or kinesin, where they like can drag along like their vesicles or whatever. They're used in the cilia and flagella as well as the centrioles. What are the tenets of cell theory? So all living things are made of cells. All cells come from pre-existing cells, and all cells are made of all cells pass down information, genetic information using DNA. What are the functions of the peroxisome? Um, it can be fatty acid breakdown, fatty acid chain breakdown, um, lipids, phospholipid synthesis, and um, it contains some of the enzymes for the pentose phosphate pathway. Microfilaments are polymerized actin filaments. They're organized into networks slash bundles in order to provide compressive resistance. It interacts with myosin in order to generate force, and it's important for the cleavage furrow. What are the functions of the centriole? The centriole can, uh, it's a microtubule organizing center. Uh, it helps to separate the chromosomes during mitosis. What are the characteristics of intermediate filaments? Um, okay, let's just see the answer. This is not good for memory, but we need to edit this probably. Um, um, so they're a diverse group of filament proteins like keratin, desmin, and lamine. Uh, withstand tension. Um, this one might be difficult to remember. What are the characteristics of archaea? You see, this is a big problem. I saw this card before, and I literally was like, "Yeah, I'm not doing this." Like, I know, but like, we saw it the other day. Like it literally showed up on UWorld. Like, that's what they wanted. Like, they actually like this is actually an important card. So, um, So it's 3, 3, 2, 1. So in DNA, the DNA is going to be bound to a protein in eukaryotes, bound to a protein, linear, and with introns. Okay. As for organelles, has a nucleus. This is like the typical definition, always the typical definition. So, um, O for ordinary. So ordinary is like nucleus, membrane bound, and then ADS. ADS isn't really that's not really like casual piece of knowledge, but okay. So eukaryotes are gonna do mitosis and meiosis, and chromosome and eukaryotes can be diploid or more. So chromosomes are paired. Average size is like 10 to 100 micrometers versus the 1 to 5 in prokaryotes. Okay, so DNA is bound to protein. I'm not looking at the screen now. DNA is bound to the protein. DNA is linear. Um, what's the last one? No introns. I mean, has introns. So DNA is bound to protein. DNA, uh, DNA is linear, has introns. Then nucleus, membrane bound, ADS. Then what was R for? R was probably for reproduction. Okay. Mitosis, it has mitosis and meiosis. And what is the last one? Chromosomes are paired. Okay. Then. Okay, so DNA is bound to the protein. DNA is bound to the protein. Uh, um, DNA is linear and it usually has introns. Then nucleus, membrane bound organelles, and the ADS ribosomes. Then reproduction, mitosis and meiosis, and and oh, the chromosomes are paired, so it's like diploid or more. And then A is what I'm gonna cover everything on the screen except for the A. Oh, the size. So it's like 10 to 100 micrometers in eukaryotes versus the 1 to 5 in bacteria. Yes. Okay. Uh, I'll see if I can remember that. I spent a long time on that card, but what are the intermediate filaments? What is their purpose? Um, the intermediate filaments are a diverse group of filaments like keratin, lamine, and some other one. Uh, keratin, lamine, I don't know, something like desmos, something like that. Uh, they can create pressure and they like help maintain the rigidity of the cell by like keeping organelles locked in certain locations. Um, and I don't know what else. Withstand tension. Cells. Wait, hold up, guys. Okay. All right. We're back now. Um, let's get the Wi-Fi back on. Okay. There we go. I know my camera's a little laggy. I don't know why, but so what are intermediate filaments? What is their purpose? Um, okay. Characteristics of archaea. Um, they're like bacteria, but they have some characteristics of eukaryotes. What are the four steps of a gram stain? So you apply crystal violet, you then apply iodine to bind it to the gram positive. Then you're gonna wash away with ethanol, and basically that's gonna clear the peptidoglycan layer from the gram negative. And then you're gonna apply safranin as a counter stain. Um, what are microfilaments? So they are uh, polymerized actin molecules, or polymerized actin filaments. They're organized as networks and bundles in order to provide compression resistance. Motor protein is myosin, and it can interact with myosin in order to generate force. It's important for the cleavage furrow. The lysosome, the plasma membrane, and the trans-Golgi network. It's gonna it covers the body. It lines the um, lines. It does line the organs, uh, and then it provides protection against infection and desiccation, and it's going to aid. It's going to be involved in secretion, absorption, and sensation. Mine, it's cavities. I said it's organs. That could be on the outside or the inside. So, yeah, I think I have to click again, unfortunately. Okay, so The DNA is gonna be linear. It's gonna be wrapped in protein for eukaryotes. Linear, wrapped in protein, and it's gonna be it has introns. Linear, wrapped in protein, has introns. Then ADS, you know what? Between eukaryotes and prokaryotes, which one is more likely to watch ads? Probably eukaryotes. Because to be honest, when have you ever seen a prokaryote watching ads, bro? You don't ever see prokaryotes watching ads. So therefore, you just got to think about ADS like ADS versus ADS. You know, so ADS ribosomes, and then we have to go into reproduction. It has paired chromosomes and um, mitosis and meiosis. And then the A is average size, which is like 5 to 100 micrometers, 10 to 100 micrometers. Yeah, I did that, to be honest. Microtubules. Um, Microtubules are gonna be, damn, bro. Microtubules are, they're hollow polymers of tubulin. Motor proteins are going to be dynein and kinesin, and they're used in the cilia slash flagella and what's it called? I think we might just finish this today, and then we're not gonna reach 60 cards today. We're gonna do the 44, and then that's gonna be it. Well, not after the 44. I think we'll still do the Jack Weston because we need to do Jack Weston. That's not even an option anymore. What are they used in slash by? Um, and then also in the central functions of the fatty acid chain breakdown, um, phospholipid synthesis, and it provides some of the enzymes in the pentose phosphate pathway. What are the components of viruses? The genetic material, obviously, but then the capsid and the envelope, which is like sometimes intermediate filaments. They're like a diverse range of filaments. I'm gonna say it's lamine, keratin, and I'm just gonna say it's like Desmond, like Desmos, but with an IN at the end. They just have three examples and they all end in IN. I remember lamine because it's in the nucleus. I remember keratin because it's your nails. But all right, so they provide they provide like rigidity pretty much in the cell, um, and like it creates. I think it helps create tension. Is it? Um, and it creates rigidity in the cell, locks some organelles in their place. Um, it's also withstands tension. It withstands tension, not creates tension. Um, it uses IF to create structures that allow cells to stick to one another. Okay. What are the characteristics of archaea? Like they look like bacteria, but they have like the genes and the pathways similar to eukaryotes. They're historically considered extremophiles because they can perform chemosynthesis, basically meaning they can use like nitrogen and sulfur to create energy. So they're gonna they're gonna cover the body and line the cavities. It's gonna protect from desiccation and infection. It's going to be involved in secretion, absorption, and sensation. What are tail fibers? Uh, Tail fibers are like, it's like the little legs, like that allow it to recognize and connect the target cells. It's a diverse range of filaments, which can diverse range of filaments like keratin, desmin, and lamine or lamin. Uh, they can withstand tension and they can withstand tension and what's it called? Withstand tension and uh, they create rigidity in the cell and what's it called? And lock like some of the organelles in their place. It also allows the cells, it creates structures that allow the cells to adhere to one another. What are tail fibers? Um, um, these are the structures on the bacteriophage that allow it to connect to and recognize its target cell. Extensions of. Okay. What are some examples of connective tissues? Um, I don't know. Goddamn. Bone, blood, bone. I thought they were putting it in alphabetical order. Might as well. I'm not gonna check if that's alphabetical order. I'm just gonna assume it is. It probably is. Uh, Blood, bone, cartilage. Why don't we just say adipose? Why are we saying fat tissue? Okay. So adipose, blood, bone, ABCs. Adipose, blood, bone, cartilage, ligaments, and tendons. See, that's so much smoother. All right, so we have our DNA is sorry, my eyes itchy. Um, so DNA is linear, wrapped around the protein, and damn, bro, this probably looks so weird. Um, uh, it's wrapped around the protein, linear, and it has introns. Then we have organelles. We have membrane organelles, a nucleus, and ADS ribosome. Um, then mitosis and meiosis, and paired chromosomes, and 10 to 100 micrometer cells. What are tail fibers? They're extensions of the bacteriophage that uh, that connect to unrecognized target cells. Um, they're polymerized actin filaments that are organized as networks and bundles in order to provide compression resistance. Motor protein is myosin, and the interaction with it allows the production of force. They generate force using its interaction with myosin. It's important for the generation of the cleavage furrow. What are some examples of? So we have adipose, blood, bone, um, cartilage, tendons, and ligaments. What are the? So they look like bacteria, but they have genes and pathways that are like eukaryotes. They're historically considered extremophiles because they are capable of chemosynthesis, basically meaning they can use nitrogen or sulfur to generate energy. Um, so they cover the body, they line the cavities, they provide protection from desiccation and infection, and they're involved in absorption, secretion, and sensation. What are nuclear pores? They're like holes in the nuclear envelope. So many of these cards need to be edited, bro. Like, okay, all right. Uh, what are intermediate filaments? Intermediate filaments are they're like a diverse range of filaments like keratin, lamin, and desmin. Their purpose, they can help to create rigidity, and they can withstand tension. Um, they can help to create rigidity and lock some of the organelles in their place. Uh, they can also hmm, now I'm not remembering that last sentence. Uh, oh, they can help the cells to adhere to one another. They create structures that allow cells to stick to one another. What are lysosomes? Um, they're fully matured endosomes with acidic environments on the inside. Membrane-bound structures containing hydrolytic enzymes. Isn't every organelle in the cytoplasm? Like that's all right. Um, I guess I guess maybe lysosomes don't really count, but I feel like it's like it's in the cytoplasm. Duh. What are nuclear pores? Um, bro, they literally are holes in the nuclear envelope, bro. That allow like an exchange between the cytoplasm and the nucleus. Like I'm not even gonna. Cristae are the fold in the inner membrane of the mitochondria. What do they help with? Um, help to create more like surface area, and like space for the ETC enzymes. The presence of the nuclear envelope permits the separation of what two steps of the central dogma. Um, uh, Transcription and translation. What are lysosomes? Um, they're membrane-bound structures in the cell um, that contain hydrolytic enzymes. It has a double membrane. Some eukaryotes have cell walls. Do eukaryotic cell walls have peptide glycan? Um, no. Even though I got that right, actually, nah, I'm gonna click hard. Nah, I gotta say again. What are the main nine differences? They they have the main nine differences are, so eukaryotes are gonna have linear DNA. They're gonna be wrapped around the protein. They have introns. That's first three. Then we're gonna have nucleus, membrane-bound organelles, and ADS ribosomes. That's second three. Then we're gonna have mitosis and meiosis, as well as uh, mitosis and meiosis, and then paired chromosomes. That's the third two. Then we're gonna have 10 to 100 micrometer sizes. Boom. What are cristae? They're folds in the inner membrane of the mitochondria. They help to increase surface area for the to make space for the ETC enzymes. The presence for of permits translation and transcription. What are some examples? Adipose, blood, bone, uh, cartilage, ligaments, and tendons. No, only bacteria. In what ways are archaea similar to eukarya? Um, okay, this is gonna be very like hyper specific, but they start translation with met, contain similar RNA polymerases, and associate DNA with histones. Okay, I'm gonna simplify this card a little bit so that so that I don't fixate on the wording. Okay, cool. Again, in what way are archaea similar to eukarya? To bacteria? Oh, to bacteria. Um, okay, okay. So a singular circular chromosome division by binary fission or budding, and similar overall prokaryotic structure. In what ways are they similar to eukarya? They have DNA with histones. Um, they start transcription with MET, and what else? Start translation with MET. Damn. Start translation with MET. Similar RNA polymerases and DNA with histones. They're like a diverse range of filaments like keratin, lamin, and desmin. Uh, they're going to they're going to withstand tension and provide rigidity to the cell by provide rigidity to the cell and lock some organelles in their place. They're also going to help create structures that are going to stick cells to one another. Cell fibers are extensions of bacteriophages that help recognize and attach to the target cell. What ways are archaea similar to bacteria? Um, so they have a similar overall prokaryotic structure. They have circular DNA, like one singular circular DNA, and I don't know what else. Oh, binary fission. Um, in what ways are they similar to eukarya? Um, they're gonna have um, DNA with histones. They're gonna have similar RNA polymerases and one more. Start translation with MET. Oh my God. What are the functions of the epithelial tissues? So it's gonna cover the body. It's gonna line the cavities. It's going to um, line the cavities. Um, it's going to provide protection against desiccation and infection, and um, the last one involved in absorption, secretion, and sensation. Yo, if you were watching me study, like you would probably see my alpha waves like just randomly spike, like in the middle of a sentence, it just randomly spikes. We haven't learned about this in the card, but my default mode network could just be randomly turning on sometimes, bro. What are lysosomes? Lysosomes are single membrane bound structures containing hydrolytic enzymes. In what ways are archaea similar to bacteria? They have a singular circular chromosome. They have a similar overall prokaryotic structure, and else. Oh, they divide by binary fission or budding. No. What are characteristics of archaea? Um, well, they look similar to bacteria, and they have genes and pathways similar to eukaryotes. They're historically considered extremophiles because they're capable of creating energy, um, using like atoms like nitrogen or sulfur, uh, or aka chemosynthesis. In what ways are they similar to archaea, eukarya? They have similar RNA polymerases. They start translation with MET, and and damn, the DNA was histones, bruh. I just gotta think like transcription and translation realistically. I feel like if I think about that, like I'll like it'll be enough. Like I'll remember. What are nuclear pores? They're literally just holes in the nuclear envelope. I'm not gonna remember the exact definition because I don't need it. What do they allow? They allow for a selective membrane between, or selective exchange between the nucleus and the cytoplasm. How's the DNA of the prokaryotic cells? They have like a nucleoid region. Yeah, that's what I said. How does the environment in the nucleus compare to the environment of the rest of the cell? Um, well, it has a lot more. Like, I would assume it would have the enzymes. It's gonna have like transcription factors. Um, how does the lysosome digest something? Uh, it would probably intake it, like phagocytosis type shit. Oh my god, I was right. Um, well, actually, hold up. I think I said that too early. Let me look this up because it's kind of confusing. Um, Yeah, I figured it was wrong because like it was just like it doesn't. I don't think it doesn't inject. It doesn't inject the hydrolytic enzymes. I don't even need a summary. Acting like it's a paragraph long too. Why is a summary needed? I don't need that. It's similar to eukarya. I better not finish at 3 in the morning, bro. I better not. 3 in the morning for the next 20 cards that most of which I'm familiar with already. In what ways are archaea similar to eukarya? Um, Archaea are similar to eukarya in that um, the DNA are wrapped around histones. Um, Translation starts with MET, and they have similar RNA polymerases, double. So the DNA is linear. DNA has histones. Um, the DNA usually has introns. Then we have a nucleus. We have membrane-bound organelles and 80S ribosomes. Then we have mitosis and meiosis and paired chromosomes. And then we have 10 to 100 micrometer cells. It's separated. It has like a selective membrane. Cristae are like folds in the inner membrane of the mitochondria that help with, they're going to help with providing enough space for the enzymes of the ETC. Translation and transcription, adipose, blood, bone, connective, no, adipose, blood, bone, cartilage, uh, tendons, and ligaments. In what ways are archaea similar to bacteria? Um, they like they have an overall pretty similar prokaryotic structure. They have a single circular chromosome, and they divide by binary fission or budding. It just fuses with like a vesicle or whatever. Um, and because it has the hydrolytic enzymes, it literally just digests it, bro. Diverse range of filaments like keratin, desmin, and lamin, and these are going to generate. They're going to be able to withstand tension, which be able to withstand tension. So going to be able to withstand tension. Um, and provide rigidity to the cell by and anchor some organelles to their like place. Um, they're also gonna help create structures that adhere cells to one another. Um, well, it depends on need. So, like a muscle cell, for example, might need more mitochondria than I don't know any other cell. How do prokaryotic ribosomes contrast with eukaryotic ribosomes? 70S versus 80S. So there's a 20S difference realistically. there's no way you need to know those specific types of RNA because like I feel like if you learn the math, maybe it's possible to learn those specific ones, but it's a 20s difference, although it's only a 10s difference. It's a it's a it's hard. It's hard to conceptualize. So basically, so we have the 70s in the we have 70s in prokaryotes and 80s in eukaryotes. The 80s are gonna add up to 70s, and like it's gonna be the like biggest possible multiples, I guess. Um, Starting at 20. So we're gonna have 80s is gonna be 50 and 20, right? Oh, okay, bro. Damn. I guess not. That doesn't work. Let me see. How can I how can I work with these numbers? Four, five, three, four, five, six, seven, eight. So if we write them like this, three, four, five, six, seven, eight, zero, 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 zero. Yeah, sometimes you just gotta like do a little rearranging. You know what I'm saying? All right, let's see if I remember that. How do the concentration of organelles in different cell types vary? Um, uh, um, yeah. So if you're like a liver cell, then you're gonna have a lot more like roughly R and Golgi. If you need a lot of energy, you're gonna have a lot more mitochondria. If you're doing like transport, like you're gonna just be lacking a lot of the organelles. What are lysosomes? They're single membrane bound structures with hydrolytic enzymes. No. Archaea, their translation starts with met. Um, they have similar RNA polymerases, and they have DNA with histones. So prokaryotic ribosomes have 70s, which is 30 and 50s, which break down to 30 and 50s. Eukaryotes have an 80s, which is 40 and 60s. Um, they just use their outer membrane. Like yes, but I'm gonna click again. So I have the cell walls in gram negative. Um, it's stored in like the nucleoid area or nucleoid region. Um, Lipoteichoic acid. Forgot about this one. So okay, so the we have the normal cell membrane. then periplasmic space, then peptidoglycan, and lipoteichoic acid, which is not shown. Okay. How do bacteria use the electron transport chain without mitochondria? They just use the space between their cell membrane and their cell wall. Describe positive sense viruses. So in gram-negative bacteria, they have uh, they have lipopolysaccharides, right? Um, there's like they're like so lipopolysaccharides. I'm trying to think of some way to remember that. Um, uh, I have no clue. Polysaccharide is like maybe like a sugar. And what does sugar do? It washes away. And where does it wash away? In gram staining. Mm, perchance. That connection genuinely might get me to remember the answer. Describe the cell wall. So um it has obviously its cell membrane. And then, but beyond that it has like the periplasmic space and then the peptidoglycan layer and lipoteichoic acid. Describe positive sense viruses. They're basically their RNA is like mRNA. Like it can be translated directly by the ribosomes. Negative sense, they cannot be translated directly by the ribosomes. They would need like an RNA polymerase to go along with it in order to basically create the complement, and then that's basically positive sense, and it can be run through the ribosomes. Oh, RNA replicase, not RNA polymerase. Um, so um, the cell walls are gonna be what's it called? Um, cell walls are um, damn, bro. The cell walls are gonna be the peptidoglycan, the thin layer of peptidoglycan, um, and then that's gonna be studied with lipopolysaccharides. Oh, it's an outer membrane that's studied with that. How does the environment in the nucleus compare to the environment of the rest of the cell? Um, it's completely separate. It has like a double membrane and a selective. It fuses with it, and then it just uses hydrolytic enzymes to digest. Negative sense is like mRNA. It can be directly translated. Oh man. In what ways are archaea similar to bacteria? They can, they are organizationally, organization, like they're very similar to prokaryotes. They also have a single circular chromosome, and then they divide by binary fission or budding. Describe the cell walls. Um, uh, Describe the cell walls. Um, Describe the cell walls. Um, Describe the cell walls in gram-negative bacteria. So they're gonna have the thin layer of peptide glycan above their cell membrane, and then they're gonna have an outer membrane that's studded with lipopolysaccharides. Describe negative sense viruses. They need to be like the viruses that carry negative sense RNA. What they're gonna do is they need to carry a RNA replicase in order to create the complementary strand. Um, and the complementary strand is positive sense, so therefore it can be translated. Describe the cell wall. So it's going to be the cytoplasmic membrane, then the periplasmic space, then lipopolysaccharides. Not lipopolysaccharides, the peptidoglycans, and lipoteichoic acid. Describe positive sense viruses. They can be directly translated. Um, they they start translation with MET, have DNA with histones, and uh, they have similar RNA polymerases. If your if your purpose is transport, then you don't need a lot of stuff. If your purpose is like secretion, then you're gonna have more RERs and Golgi. Uh, if you need a lot of energy, you're gonna have more mitochondria. So prokaryotic ribosomes are 70S, so it's gonna be 30 and 50. 30 and 50S, and then eukaryotic are going to be 80S, so it's going to be 60 and 40S. How do bacteria use the electron transport chain without mitochondria? They use the intermembrane space. Describe the cell walls in gram-negative bacteria. Um, they're going to be they're going to basically consist of um, so you're going to have the periplasmic space that's going to have the peptidoglycan, and then you're going to have an outer membrane that's going to be studded with lipopolysaccharides. The cell wall um, is going to be cell wall and gram-positive cells is going to be the uh, lipoteichoic acid and polysaccharides. What? Why would I say polysaccharides, bro? Oh my god. Describe positive sense viruses. They can be directly translated. The cell one gram positive, it's a thick layer of peptide glycan and lipoic acids, bro. Describe negative sense viruses. They cannot be directly translated. It means that they commonly carry RNA replicases in order to create the complementary strand that can be translated. In gram positive, they're going to have a thick layer of peptide glycan and lipoic acid. All right, now let's do some Jack Weston and go to sleep. All right, let's get this. Think number one, we're finally done with bio. I'm so glad. Uh, right, let's come over. Uh, over here. Then check Western. to stop here. This was like the worst passage. Damn. Damn, I just clicked the random one. I wasn't sure. It was just because of the connotation, like the way that they said it was like war. It's war without the shooting. Like without the shooting, like at that point you're just using it for its connotation. This one was kind of hard. I wasn't sure about this at all. It's this last question that I really want to see. Bro, there's no way this was an easy question, bro. 80% of people got this right. What? But how would you know? like, it's so that's so like random. It's like actually so random. I wouldn't have expected that. It's like Damn. I didn't know I didn't know it was 50-50. Damn. Okay. So I pretty much got that average. Damn. That's unfortunate. Okay. All right. Well, everyone, I hope you have a good night, good evening, good afternoon, and any other part of the day that you would like to have a good version of. I'm gonna go. It's time for me to go to sleep. But good night. Bye.